So the title is as introduced. And then before talking about the main story, I'd like to mention about myself a bit because my background is different from most of you sitting here. I got a PhD in engineering, computer engineering, and then even if I came from different area, I got interested in biomedical applications a lot. So even when I was a PhD student, I was dealing with so many different types of biomedical data sets and applications and topics and issues. So I've been working on this area for 15 years. Then 2 years ago, when I joined as a faculty member at Kyunghee University School of Medicine, I found a great team at the Korean Medicine Acupuncture, the hospital. As you can see here, Dr. Baek, who just introduced me, and Dr. Seo and Dr. Park. So this is our collaboration. So in this collaboration, the three MD PhDs are focusing on mainly clinical studies and researches, and then specifically for this collaboration, they are collecting patient data sets. And then we have another team, Dr. Lee's group here. He's going to talk about his stuff, right after me. So he's focusing on animal model studies using zebrafish. So he's been doing, developing many different types of experimental approaches, and then he's mainly doing so many different experimental validations as well. And then my team in the center, we are in computational systems biology, and I've been developing many different in silico simulation models and approaches and pipelines. So simply saying that I've been working on data-driven analysis and data-driven predictions using biomedical data sets. So in this collaboration, before starting a few projects, we had a question like this. Started from there. Korean medicine is already well known. People love it. People love to get the treatments. And then but the thing is, do we already know what's going on for the functional mechanism, basic functional mechanism? Simply saying that, do we know what's going on inside cells for the treatments? Maybe not. I know some tried, but not enough, maybe not sufficient. So in this topic, cold or heat pattern of 사상 symptomatology is mainly determined by clinical information, as far as I understood. And then the following treatment is determined by the characteristics of the original symptoms. And then we still don't know what kind of functional mechanisms for the treatments. That's how that's why I'm here to use our techniques and skills and know-hows to get to know what's going on in there. So simply saying that we want to know what's going on inside cells. As you may be already aware of, inside a cell, there are so many different types of molecules. And then people already classified them into 4 different system levels. Genome level, DNA, and transcription level, RNA, and protein level, protein, and metabolic level, metabolites. And each system level it's got its own characteristics, and then it's being operated, with its own ways. And then at the same time, they are interacting with each other, closely. So I've been developing many different algorithms and then pipelines to get to know what's going on in each system level. And at the same time, recently, I've been dealing with many different approaches combining those interactions. So for this project, cold or heat pattern, Dr. Park has been collecting blood samples from individuals, 25 individuals. And then I might say sorry for this example. Did not have enough time to translate them into English. But here you can, anyone who's got interested in, you can take a look at the paper here, down here. Dr. Park collected blood samples based on this survey and got some scores. We have, is it working? Yeah. We have two different scores. Score for heat, it's got a range between 7 and 35. And score for cold, it's got range between 8 and 40. So for example, to have a cold pattern, the score for cold should be higher than 23.5. And then at the same time, the score for heat should be lower than 20.5. Then we might say this one can be classified into cold pattern. That's how we collected 25 samples. Then we sequenced the blood samples to get RNA-seq data sets. So RNA-seq data set, it's got transcription expression changes. So we can get to know, using the data sets, we can kind of guess, what's going on at transcription level. And then previously, I developed a in silico metabolic flux simulation model. So using the model, we can kind of guess what's going on at metabolic level as well. So we are, in this project, taking a look at two different system levels. So using the RNA-seq data sets, we first didn't consider whether it's a male or female. We just took a look at the difference between cold and heat pattern, and then got some significantly differentially expressed genes, and then found out the top list of the genes came from sex chromosome. So decided to get them separate, like this, male and female. And then again, got the significantly differentially expressed genes again, like red dots here, and took a look at the similarity, and then found that not many common genes are there. So we kind of conformed to go further this way. Then using those significant differentially expressed genes, we were curious about what kind of known signaling pathways were associated with them. So we performed one of basic analysis called gene ontology analysis, then got some list of known signaling pathways like this. For male, we had a lipid metabolic process or protein localization or like small molecule metabolic process and so on. And then for female, this came from comparison between cold pattern and heat pattern. And then for female, we got like biogenesis and then protein modification and again biogenesis and so on. We have another metabolic process here. But based on this analysis, we could not have enough information to go further to decide to go this way or that way, So we did again another analysis called gene set enrichment analysis. So again, we were trying to get to know what kind of known signaling pathways are associated with. From this point, I made all the blue colors as for cold pattern, and then all red colors are heat pattern. So here we have for male, we have all the blue colors, which means it's got alterations. More alterations in male with cold pattern compared to male with heat pattern. And then we found some, not just some, many of them we found that were associated with so many different metabolic processes. Here you see a glucose metabolic process, carbohydrate metabolic process. And then we have some other catabolic process as well. Where is it? Yeah, here. And then metabolites and energy. So that's what we saw in male. And then for female, now you see more red colors, right? So which means female with heat pattern showed further alterations compared to female with cold pattern. And then we got some consistent, findings. Sorry. Like from the center, you can see lipid metabolic process, steroid metabolic process, and then some cellular catabolic process, and so on. So based on those analysis, we decided to go further to metabolic alterations. So in order to get to know what's really going on for metabolic changes, we took a look at gene expression profile changes for specific pathways associated with energy metabolism. Because energy metabolism is the primary one for the metabolic changes, alterations. And then here we used a gene list provided from Dr. Douglas Wallace at UPenn, one of my collaborators. He's actually one of pioneers in mitochondrial energy metabolism research. So this pathway is oxidative phosphorylation, the main pathway for energy metabolism. And if you take a look at the male first, you can see here now Z-scores, which means relative expression levels, like up or down, just binary. And you can see some of them up, some of them down. It doesn't really show a clear pattern between cold or heat pattern. But here for female, you can see most of them are upregulated in female with cold type, which means it may have like a more like association with energy metabolism. And then we also took a look at other pathways as well associated with energy metabolism. Again, here we still see Z-scores. So by looking at these colors, you can see up or down, still binary. And then here we provided another score, which is called the T-score, which means if you see any dark red or dark blue, which means it's got significant changes. So let's take a look at some pathways. Energy sensing kinase and mTOR pathway, we have seen upregulation on male with cold pattern. Again here, upregulation for male with cold pattern. Interestingly, fatty acid oxidation, one of main pathways for energy metabolism. It's got upregulation again for male with cold pattern, and then female with cold pattern. However, if you take a look at the TCA cycle, some of them got up, some of them got down. So it doesn't really give clear pattern. So this is what we got for the first one, and then the second page. We still have pathways directly associated with energy metabolism. So glycolysis. Again, unfortunately, it doesn't really show a clear pattern between heat or cold. And then for mitophage, based on this pathway, we can kind of guess, how mitochondria are being used efficiently or effectively. However, again, it doesn't really show clear pattern between heat and cold. And then one more thing, let me talk about, which is antioxidant defenses. If you take a look at this one, it's not 100%, but female with cold pattern has got upregulation for antioxidant defenses. And then one more pathway we need to check. This one, stress response. Again, it's got upregulation for female with cold pattern. Those two pathways are associated with oxidative stress, which means the female with cold pattern might have more oxidative stress. And then one more thing. Let's take a look at nucleotide synthesis. So as I told you, it's got if it's got dark red, it's got significant alterations. And then here, if you take a look at this part, and then female with, again, cold pattern, it's got upregulation for nucleotide synthesis, which needs lots of energy for the synthesis. So in a nutshell, let me briefly summarize what we have done, what we have seen. Female with cold pattern might generate more energy. At the same time, might spend more energy for those kind of stuff, like a nucleosynthesis. And then, while doing that, it may have more oxidative stress. So that one should be an interesting point. And then we also took a look at gene expression profile alterations on CAG map. So we can take a look at where it's got up or down. So as we have already seen, heat map of TCA cycle, as you may remember, it did not give clear pattern. As we can see here, it doesn't really give a clear pattern between heat and cold, but it shows definitely different pattern between male and female. Here you can see red, and then here you can see more blues. And then again, oxphos. As you may remember, female with cold pattern, it's got upregulation on oxphos, right? So we see many of them as blues, right? And then another interesting point I would make is from the heat map, we didn't really see clear pattern for male for oxphos, but here, so some of them got up in blue, some of them got up in red, but if you take a look at the complex 1, 2, 3, 4, 5, the main, main road for the energy generation, all red colors you can see here, which means male with heat pattern might have something, but I didn't dig into further yet. And then other pathways like glycolysis, as we saw, it did not really clear pattern from the heat map. And then here we can see, some of them got up in blue, some of them got up in red, but it doesn't still show clear pattern. And then, since we got interested in metabolic changes, we again implemented another GSEA approach using another package, FGC, which is called FGC, with specific gene list called Mitocarta 3.0. So in this analysis, we can kind of guess what kind of alterations it's got based on metabolic point of view. Here, again, red color is associated with a heat pattern. So for male with heat pattern shows upregulation on calcium-associated pathways, calcium homeostasis, calcium cycle, calcium unipolar. In other words, male with cold pattern has got downregulation on these calcium-associated pathways. Again, in other words, male with cold pattern might have more mitochondrial dysregulation. And then this one is consistent with what we have seen so far. So female with cold pattern, it's got up, right? And then female with cold pattern, it's got up in mitochondrial biogenesis. And then we've also taken a look at, many different metabolic pathway alterations as like this. You can take a look at, some of them are associated with energy metabolism, and some of them got associated with lipid metabolism. And then this one is the highlight. So as I mentioned, we previously developed in silico metabolic flux simulation, so we can take a look at what kind of differences, what kind of alterations it's got on metabolic level. So this one is one example for carbohydrate metabolism. Here you can see glycolysis, TCA cycle, pentose phosphate, pyruvate metabolism. And then if you compare cold and heat, by the way, in this simulation, we can kind of guess activation level of all the known metabolic reactions. So those metabolic reaction activities are presented in this color scale. So you can directly compare using the colors. Let's take a look at the differences. Here, we can definitely see the difference, right? So this part is the difference in glycolysis, and then this part, this reaction's got alterations between heat and cold, and then this part, it's got alterations. And then for female, again, this part, it's got alterations. And then again, this part, it's got alterations for glycolysis. And then again, here, this one. And then not really clear, but the color scale is a bit different in pentose phosphate, part of a pentose phosphate as well. So something like that. We can compare those kind of reaction activity levels. And then we can also take a look at them as heat maps like this. So as I told you, the metabolic alterations got associated with either mainly energy metabolism or lipid metabolism. So this is one of our lipid metabolism example, fatty acid synthesis. So for male, it's got a clear difference, upregulation in male with heat pattern, right? But for female, it does not really give a clear pattern. Some of them got up in here, some of them got up in there, something like that. So these are two different views to take a look at, what kind of alterations it's got on metabolic level. And then here you can see we had only 2 samples for a male with a cold pattern, and then only 2 samples for a female with heat pattern. So we're working on it. We have added more samples, and then as far as I know, it's being processed for RNA sequencing. So as soon as we get more samples, and then we're gonna have a clear conclusion, and then hopefully you can take a look at the paper early next year. So this is it. I'm always thankful to my lab members, and then thank you for listening, and any questions you have, please. Thank you.